DNA replication in prokaryotes and eukaryotes. DNA replication is semi-conservative and requires a template. Deoxynucleoside triphosphates (DNTPs) are the raw materials or the precursor molecules for the DNA synthesis. Nucleotides are added only to the three ends of the growing nascent chain. Therefore, the nascent chain grows only 5 to 3 direction. Addition of nucleotides to growing chain called the chain elongation. Addition of nucleotides to nascent chain is catalyzed by the DNA polymerases, equally consisting of the three different DNA polymerases in the form of DNA polymerase 1, 2 and 3. DNA polymerase 1 first discovered in 1950s by A. Korenberg. It is having the three different enzymatic activities in the form of the 5 to 3 polymerase activity for elongation, 3 exonuclease activity for proofreading, 5 to 3 exonuclease activity, it removes the RNA primers. Without 5 to 3 exonuclease activity in the DNA polymerase 1 is known as the clinose fragment. It is useful in case of the DNA sequencing. A. Korenberg, who discovered the DNA polymerase 1. 3 exonuclease activity, which is useful in case of the polymerase 1 for the proofreading. 5 exonuclease activity, it removes the RNA primers. In 1960s, biologists thought that more than one DNA polymerase is existing equally, and in 1970s, polymerase 2 and 3 were discovered. The DNA polymerase 2 and 3 do not have 5 to 3 exonuclease activity, but they have 5 to 3 polymerase activity for elongation, 3 exonuclease activity for the proofreading. DNA polymerase can only elongate existing chains, it cannot initiate de novo chain synthesis. Nascent strand initiation requires the formation of the short RNA primer molecules. The RNA primers are synthesized by the RNA primase, a type of 5 to 3 polymerase capable of initiating nascent chain synthesis from DNA template, uses ribose NTPs as nucleotide sources. 5 primers, the primers are eventually excised by the 5 exonuclease activity of the DNA polymerase 1. Replication begins at the location of the chromosome is known as the ORI site, origin for replication. DNA helix unfolds forms the origin 2 old strands in the form of the 3 to 5 template and 5 to 3 template. Replication on 3 to 5 template, the DNA synthesis will takes place in the 5 to 3 direction in a continuous fashion. It is known as the leading strand. On the 5 to 3 template, the DNA polymerization will takes place in the 5 to 3 direction in the form of discontinuous way, with the each discontinuous strand is having RNA primer. It is known as the lagging strand or Okazaki fragment. A primer excision is completed by the DNA polymerase 1 and it will be fill the gap and by whatever the nick is stick together by the ligase enzyme. DNA polymerase 3, which is one of the principal DNA duplicating enzymes, it synthesizes both the leading and lagging strand by using the 5 to 3 template. So on the 3 to 5 template, the DNA polymerase, which is synthesizing in the form of the 5 to 3 polymerase activity by using the alpha subunit, one of the core enzyme. Another core enzyme which is synthesizing the 5 to 3 template by the 5 to 3 polymerase activity in the form of the discontinuous way, that is known as the Okazaki fragments. So the DNA polymerase, which won't synthesize on its own the nascent strand. For that we can see the one of the primase enzyme which synthesizes the RNA primers on the template DNA. After that, it will be handed over to the DNA polymerase III. Whatever the RNA primers will be digested by the RNase H or DNA polymerase I, which removes the RNA primers by using the 5-3 exonuclease activity, and the gap is filled with the 5-3 polymerase activity. Whatever the nicks are there in between two DNA fragments will be sealed together with the ligase enzyme. The two DNA strands will be unwinded by using by using helicase enzyme, which is synthesized by the DNA B gene. With the help of the ATP, they will be separated in the form of the two strands. We can see in the DNA duplication different enzymes in the form of the primase, DNA helicase, single strand binding proteins, and topoisomerases. Primase, which is synthesizing by DNA G, or primase enzyme, which is synthesizing the RNA primers. DNA helicase, DNA B gene, which separates the two DNA strands in case of the prokaryotes. MCM complex in case of the eukaryotes. Single-stranded binding proteins, which binds the single-stranded DNA in case of the prokaryotes. In eukaryotes, replication protein A will bind to the single strands. It keeps the single-stranded status not to self-annul. Topoisomerases, which makes the nick on the single-stranded DNA, and it removes the supercoils. Whenever the DNA duplicating at one region, another region supercoils will be formed. These supercoils are removed by the topoisomerases. DNA gyrase makes a temporary single-stranded nicks in one of the two template strands, relieves the torsional stress and supercoil caused by the unwinding helix. And we can see the single-stranded binding proteins, which stabilizes the single-stranded status long enough. We can see the different DNA enzymes in the DNA duplication in the form of the topoisomerase, 2-gyrase, DNA gyrase, which makes the single-stranded nicks, removes the supercoils. DNA B gene, which is the helicase enzyme, which separates the two DNA strands. DNA primase, which synthesizes the RNA primer for the synthesis of the new strand on the DNA template. And we can see the DNA polymerase III, which synthesizes on one of the strands that is continuously, that is known as the leading strand. On the other strand, which is synthesizing in the form of the short fragments with RNA primers, they are known as the Okazaki fragments. And we can see after the DNA duplication, in E. coli, the two ring-shaped DNA molecules will be separated by topoisomerase II. And over the view of the DNA polymerase in the bidirectional origin for replication, we can see a continuous DNA synthesis on leading strand, discontinuous way in case of the lagging strand. In eukaryotes, the linear chromosomes which have the telomeres at the ends. The telomeres will protect the chromosome lengths in the eukaryotes. If the chromosomes of the germ cells become shorter, every cell cycle essential genes will be missing, and an enzyme called telomerase catalyzes the length of the telomerase in germ cells. In case of the telomerase, we can see one of the enzyme telomerase, which is having one of the RNA template and reverse transcriptase enzyme. It extends the DNA length in the 5-3 strand, and by synthesizing the 5-3 way. In this, we can RNA template and telomerase enzyme, which extends the 5-3 region of one of the strand of the DNA in the form of the TTA triple G-like structures in the form of the repeats. These are the three persons will recognize the telomerase enzyme: Blackburn, Grider, and Sojastock. Eukaryotic DNA replication. Eukaryotic chromosomes have multiple origins for the replication. We can see that there are 6 different eukaryotic DNA polymerases. Alpha, which is doing primase activity, which is having only 5 to 3 polymerase. There is no 3 to 5 exonuclease activity for the it is having only 5 to 3 polymerase activity, but there is no 3 to 5 exonuclease activity for the proofreading. In addition, we can see the epsilon and delta, which are having the elongation leading strand epsilon, delta, which is useful for the DNA duplication on the lagging strand. Beta DNA polymerase, which is useful for the repairing mechanism. Gamma DNA polymerase only in case of the mitochondrial replication. DNA polymerase switching during eukaryotic DNA synthesis. The primase enzyme which synthesizes the RNA primer, it is extended by the DNA polymerase alpha, which is synthesized in both leading and the lagging strand. It is only having polymerase activity of 5 to 3. There is no any proofreading of the 3 to 5 exonuclease activity. We can see the replication fork C and polymerase proliferating cell nuclear antigen, which is useful for the DNA synthesis by the help of the epsilon, which is synthesizing on the leading strand in a continuous way. Delta on the lagging strand, it is synthesized in a discontinuous way. So these are the 3 scientists in Roswell and Franklin, Barbara McClelland, L. Blackburn are the women scientists. We can see in the DNA polymerization in prokaryotes and eukaryotes. In case of the eukaryotes, we can see there are 2 different DNA polymerases in the form of epsilon and the delta, which epsilon which is synthesizing on the leading strand, delta which is synthesizing on the lagging strand. In case of the eukaryotes, the RNA primers will be removed by the RNase activity and also FAN1, and the gap is filled by the delta DNA polymerase. In prokaryotes, the RNA primers is removed by the 5 to 3 exonuclease activity of the DNA polymerase 1, and the gap is filled with the DNA polymerase 5 to 3 polymerase activity, and proofreading is done by the 3 to 5 exonuclease activity. So like this we can see the RNA primers will be removed in prokaryotes and eukaryotes in a different way, whereas delta which is filling the gap in case of the eukaryotes, whereas DNA polymerase 1 which is filling the gap in case of the prokaryotes. After that the nick is filled with the ligase enzyme. So like this we can see the different DNA polymerizing enzymes which are synthesizing in the prokaryotes and the eukaryotes. In addition, in case of the eukaryotes, we can see in addition to primase enzyme, we can also see the alpha DNA polymerase, which is also doing the primase activity. In addition to RNA primer, we can also see the DNA primer, which is synthesizing both leading and the lagging strands. So, but the alpha primase do not have the 3 to 5 exonuclease activity proofreading, but it handovers the respective DNA strands to the epsilon for the leading strand and delta for the lagging strand. So altogether the DNA duplication will be takes place in prokaryotes and the eukaryotes by using the different DNA polymerases, and we can see the protection of the chromosome length by the eukaryotes in case of the telomerase enzyme, so that keeps the chromosome length intact.